everybody put your cell phones away. I haven't said that in a while. So I have really good news I think you're all going to love. Tomorrow you won't get sandwiches. But on short notice, all I could do was pizza and a salad bar, but it is not sandwiches, so we should be thrilled. And we're hoping that we can have something that's not sandwiches every day for the rest of the week. I knew this would make you guys really excited. Unfortunately, today you have sandwiches.
So you'll notice that I sent out another email for final reflections. Those are due at the end of the week. Those are due date there. But a reminder that I think some of you guys are working on team videos. There will be an official request going out for that later today, as well as a final course evaluation. So it's important for us to get the feedback from you guys about how we're doing, how the courses are going, and how you think you guys are growing over the course of the work. So thank you for doing out those evaluations. It's from a team point of view. I want to see how the teams are working together. You should just, whatever the team feels like they should be doing, they can talk about. So, and show how you're making new friends. It's anything. So, thank you. And here's Danelle.
I really don't like microphones. Hello. Happy Monday, everybody. So I've heard that your favorite part of the day is yelling out when we do the course attendance, right? So today, when I call your course name, cheer loudly. So I would like it to be proportional to your excitement to the fact that we are not going to be having sandwiches for the rest of the week, right? So if you sound really sad, we can make it so that you just have sandwiches. So for those of you who don't know me, I'm Danelle. My supervillain name is Dr. Shaw. I am instructing the Medlytics course this summer. Yeah. All right, so we already know my Medlytics students are here. Give it up. Yeah. All right. And I vowed that I wouldn't forget anybody. So Cogworks. All right. Embedded security. Sounds like they want sandwiches. That's what I hear. 3D printing. CubeSat. UAV. All right. UAV SAR. And race car. All right, well, they have twice as many students, so. Okay, so today I'm super excited to be introducing our guest speaker today, Dr. Ed Boyden. Dr. Boyden is a Y. Eva Tan Professor in Neurotechnology at MIT, Associate Professor of Biological Engineering and Brain and Cognitive Sciences at MIT's Media Lab, and McGovern Institute for Brain Research right across the street. He leads the Synthetic Neurobiology Group and co-directs the MIT Center for Neurobiological Engineering. And today he's come to talk to us about technologies for understanding and repairing the brain. So please join me in welcoming Dr. Boyden.
Cool. Great. It's really good to be here. I trained as an engineer. In fact, I sat in this room and other rooms in this building many times myself, 25 years ago or so. And when I finished my engineering training, I decided, what do I want to work on for the rest of my life? And I looked around, and there were lots of problems to work on, obviously. But pretty much none of them were as daunting and challenging and annoying as the brain. So, how does it work? How does it implement things like thoughts and feelings, our memories, our identity? And the incredible density of computational processing of the brain really struck me as, we got to do something about this. If we can understand how the brain computes, these dense networks of brain cells called neurons, which are super dense, by the way. So in a cubic millimeter of your brain, you're going to have 100,000 brain cells called neurons and 1 billion connections between them. And each of those brain cells computes with millisecond timescale electrical pulses, and each of those connections communicates with these millisecond timescale chemical pulses. So if we could actually understand the brain, who knows? Maybe we could generate true human-like AI. Maybe we could try to understand what it means to have a feeling or a thought, or to make a decision, or to see something, or to move. But I would argue this is more than just about building true AI. It's more than just about understanding the human condition. There's some real urgent problems as well. So if you look at brain diseases, and as we make inroads in other kinds of disease, these are often some of the worst things around. If you look at the list here, Alzheimer's and epilepsy and spinal cord injury and Parkinson's disease, pretty much none of these can be cured. And the treatments that exist are partial. They don't work very well for the most part. And they affect something like 1 billion people already around the world. And I think part of why these diseases are scary is that they don't just change the time we have to live, they can change who we are, how we relate to loved ones and friends and family. So maybe not surprisingly, because the brain is so darn complicated, building therapies to treat brain diseases take forever. This is from a 2010 editorial. I think the numbers are worse by now. The failure rate to get clinical approval is over 90%. I think the numbers now for some diseases are essentially 100%. The cost is enormous, billions of dollars each. I think the latest number suggests that it's around $2.5 billion per attempt. And as I mentioned earlier, even when you do get through all these hurdles, the treatments don't really work very well. So as an engineer, I did a bit of chemistry, I did a bit of electrical engineering, I did a bit of computer science. It's useful to take a step back and say, why is the brain so darn hard to understand and fix? And I thought a lot about this for many years. And one thing that I realized was, if you look at the triumphs of the 20th century, shown in the top half of the slide in abbreviated form, obviously, we have airplanes and transistors and lasers and landing on the moon and so forth. These are all built really, you could argue, on areas of engineering that stem from physics, right? Where there's a small number of things and a small number of ways they interact. And so we could engineer things really, 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 really well. Flash forward to the 21st century. In the human genome, we have 30,000 different genes. So if you think about even a naive model where they're just on or off, 0 or 1, that's 2 to the 30,000th possible states, which is very, 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 very large. Now imagine all the interactions between these genes, and it becomes a truly terrifying spectacle. So that's why I think biology and medicine are so hard to confront. But the brain is especially difficult because you're dealing with massively differing spatial scales and temporal scales. So I showed you this video before, but now I'm going to tell you what we're doing. Brain cells are enormous, right? They're centimeters in spatial extent. The neurons that go down our spinal cord are 1 meter in length. They're by far the largest cells in the body. But if you can zoom in with a microscope, let's say, onto the smallest connections between brain cells, these synapses as they're called, or even within a synapse, the molecules, the gene products that make the brain cell do what it does, those are nanoscale. An average biomolecule might be 2, 3 nanometers in spatial dimension. So how on earth can you see something as large as a brain, centimeters, with nanoscale precision in 3D? And I'll show you how we do it in a second, but until recently, there really wasn't any way to do that. The second issue is time. So most biology systems we can kind of deal with on a slow basis. You can take an antibiotic for a week, and it fights the pneumonia, and a week is not a bad time scale for dealing with some infectious diseases, for example. But in the brain, you're dealing with really brief events. Each brain cell has millisecond timescale electrical pulses within it, and they communicate with each other with these millisecond timescale chemical exchanges. And yet, if you care about Alzheimer's disease, or brain cancer, or depression, or Parkinson's, these are phenomena that last years, decades, and develop over long periods of time. So I would argue that really our job as neuroengineers, which is what my group here at MIT does, is threefold. Number one, how do we develop a science of engineering that's centered around lots of building blocks interacting in lots of different ways? Number two, how do we cross space? How do we see a large object with molecular precision? And three, how do we cross time? How do we solve long-term things like memory or identity or Alzheimer's or brain cancer without losing track of the quantum building blocks of the computations of the brain? So the way my group works is we take one of these big challenges, like let's see everything, or let's be able to look at high-speed operations of the brain, and we work backwards from it. We survey all the different disciplines of engineering: chemicals and electrical and robotics and all sorts of stuff, and you'll see a little bit of everything in this talk. And then, can we build tools that solve sort of this tripod of goals that I'll talk about on this slide? Number one, can we map all the molecules throughout the brain cells and the brain cells throughout the networks that make up the brain? Number two, can we control the high-speed electrical and chemical dynamics of the brain? And number three, can we watch those high-speed electrical dynamics in action? So one thing we want to do, of course, is if we can watch a brain while it's computing, we watch the high-speed dynamics. We perturb it to test causal theories of how the brain works. And then finally, at the end of the day, we map the molecules and cells and structures. Could we actually simulate what the brain does in a computer? I think we've all heard a lot about triumphs of machine learning and AI in recent years, but there's lots and lots and lots of things that these algorithms can't do. And so what if we could build AIs that could do that? What if we could pinpoint how the brain changes in a disease? Could we figure out the exact targets in the brain that need to be repaired in order to end or reverse a brain disease? So for each of these three mega goals, our group has been developing a technology that, as you'll see in a second, is often quite unexpected. And that's because we're backwards thinkers. We're thinking backwards from the big problem, and then we try to survey all the different disciplines of engineering to conquer these really, really difficult mountains. Let's start with the first goal. How do you map the molecules throughout the brain cells and the brain cells throughout the networks with enough precision that you could maybe someday make a model of a brain circuit in a computer, but not lose sight of the enormous scale of the nervous system? This is an old problem. Lots of people have tried. In fact, the Nobel Prize a couple years ago was given to some of the inventors of so-called super-resolution techniques, where you can zoom in into very, very small parts of cells. But these technologies are too slow and too expensive to be applied to large-scale brain circuits. I think we've all seen pictures like this, brain scans taken with MRI scanners. These are too spatially crude to reveal the networks of the brain. So these blobs or voxels that you see lighting up are actually not neurons. They're measuring blood flow. And each blob or voxel contains millions and millions of brain cells. And even two brain cells right next to each other can be doing completely different things. At the other extreme are microscopes. You can zoom in and see small things, but not small enough. Light has a finite size or wavelength, and you can't see things much smaller than it. These super-resolution methods that I just alluded to, they tried to break that resolution limit, but again, they're really, really slow and difficult to apply to three-dimensional objects, which your brain is. So a couple years back, we started thinking, what if we do the opposite? For 300 years, people have been trying to zoom in, often using lenses, to take pictures of things. What if we blow everything up? What if we take a brain and physically inflate it until it's thousands and thousands of times bigger? Could we see all those fine connections and the molecules at those connections? But we could take the pictures using really, really cheap optics, like maybe modifications of the cameras you find on cell phones nowadays. You can make scalable imaging really, 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 really cheap and really, 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 really fast. So this idea we call expansion microscopy. And what we do is we take a piece of brain tissue, preserved, it's not living at this point, and we synthesize throughout the entire brain a spider web of swellable polymer, a polymer not unlike the stuff in baby diapers. And the answer is no, I did not think of this while changing a baby diaper, just to get that out of the way. So what if we could install these polymer threads throughout the entire specimen in an even way? And then if we add water, as kids do every day, the polymers will swell, but if we do it right, maybe we can pull all the building blocks of life away from each other. So this combines two old ideas. One is from MIT physicist Toyoichi Tanaka, who in the 80s was working at the physics of these swellable polymers. So this movie shows you what this looks like. You take a swellable polymer, you add water, water's drawn in through osmosis, and then these polymer threads are super highly charged polymer backbones. So they repel each other, like charges repel, and you get this massive volumetric increase. And so that's, in case you wanted to know, how a baby diaper works. The other thing you got to figure out is how to install the polymer inside the brain. You can't just dump the polymer on top. It'll just sit there. We tried. What you have to do is you have to weave those spider webs around and between the biomolecules. It turned out that was also figured out in the early 1980s by a couple people at the Max Planck Institute in Germany. And the answer is you take the tissue and you soak it in the monomers, that is the building blocks of the polymer, and they self-assemble to form these polymeric threads that weave their way throughout the specimen. So one big theme of neuroengineering, which is why I find it so much fun, is that you often find these really, really, really great ideas just hiding in plain sight. You know, these two ideas have been lying around for nearly half a century. Why didn't anybody bother to do it? Well, if you could do it just right, maybe you could take a brain cell, like the cell on the left, and pull all the biomolecules apart from each other. So like a constellation of stars in the sky, the biomolecules hover in space, but anchored to the polymer. And of course, they're going to be farther apart from each other, but if their relative organization is preserved, then all the information you need to reconstruct that brain cell is still there. So to make this work, we had to invent a couple new chemistries. The first thing we had to invent were anchors, basically molecules that would bind to biomolecules and apply force so we could pull them apart. And that's what you see in this cartoon. The brownish blobs are proteins, and the purplish blobs that you see wandering around are the anchors. We devised anchors that bind to the genome, that bind to proteins, and so on and so forth. And basically, they're going to equip every biomolecule with a little handle. The second thing we have to do is to weave the polymeric threads. And we do that just like the Max Planck groups did in 1981. We had monomers, shown as little white spheres here, and they form these polymer chains that wind their way around in between the biomolecules. And when they bind to one of these anchors, they're going to form a tight connection. And so if you think about it, the polymers that you saw swelling earlier can provide force, and the anchors can convey the force. This is like robotic engineering at the nanoscale here, except we want our robots to infiltrate everything. And the final step is we have to loosen everything up, so we can use detergents and heat to loosen up biomolecules from each other. And then when we add water, just as Tanaka had figured out half a century ago, the polymer threads will swell. But this time, the biomolecules come along for the ride. So here's a piece of brain tissue from a mouse in panel B. And in panel C is the same piece of brain tissue, but some number of hours later. And it's grown 100 times in volume. And the polymers go from something like in the upper left to in the lower left. We think they kind of expand in this very even fashion because they're really dense and really evenly made. So here's a little movie of a piece of brain growing before your very eyes. It was polymerized earlier, and then we add water right there. And I hope what you can see is that it's actually expanding. I'm sure it's only a matter until some Hollywood scriptwriter makes a horror movie out of this. Revenge of the Growing Brain. So the whole process takes like an hour, not a minute. It's sped up in this little time-lapse video. But the point I'm trying to make is that we can actually blow up brain tissues evenly because the polymers are so dense. The spacing between the polymeric threads is around the size of a single biomolecule. So what can we do with this? Well, as I mentioned in the beginning, the brain cells are connected by these nanoscale connections that you can't see. And the wires are also nanoscale, and you can't see them either. So now we can do things like expand the brain and take pictures. So here's a part of the brain, this is also in mice, called the hippocampus. It's involved with many things, including memory formation. So if you lose the hippocampus, you can't form new memories, although you can still remember old ones. So it's been a big mystery. How can a part of the brain be sort of a scratch pad, like RAM in a computer, for short-term memory, and how does it actually encode things? Well, we took a piece of the brain and we color-coded the neurons. You can do that by using viruses. The way it works is you bring in a virus, and different brain cells will take up different viruses. And each virus could have a different color inside of it. That's because there are proteins in the wild that glow different colors, and you can load different proteins from different species, like from coral and jellyfish. The coral protein is red, the jellyfish protein is green. Anyway, you can take these molecules from different species, put the genes in viruses, and if different brain cells take up the different viruses, they'll have different color codes. So that was invented by a group at Harvard. They call it Brainbow. It yields beautiful pictures. But if you zoom in to one of these Brainbow brains, it's fuzzy, because microscopes can't see things much smaller than light. What's this green banana shape, for example, up at the top here where my mouse is pointing? Who knows, right? That's before you expand. But after you expand, you can clearly see the green banana shape is not a green banana. It's got very distinct wires, 1, 2, 3, 4, and you can tell them apart. So can we actually start mapping the brain? It seems that we can. We're actually now doing things where you expand a brain and then try to image all the synaptic connections in an entire circuit. This is some work we've been doing with Eric Betzig, who's actually one of the people who won the Nobel Prize a couple years ago for his work on super-resolution microscopy. If we expand and then use modern microscopes to image, we can go 1,000 times faster than the best previous method. So here's an example where a mouse, we have neurons expressing a fluorescent protein. Here's the entire thickness of the cortex, which is one of the most interesting parts of the brain, involved with all sorts of stuff, from decision-making to memory recall and everything in between. And these little squares here, we're going to zoom into. So this box that says 2, We're going to zoom into it here, and this little box here, and we're going to zoom into it further. And what you can see are we can get down to individual connections. But again, these are all part of a giant, giant image that we can zoom out and see the entire network. So here's actually a little movie of us flying through one of these little boxes. Yellow are the brain cells that we've labeled, and then magenta and blue are the different parts of a neural connection, the so-called pre- and postsynaptic sides. And so really for the first time, we can image an extended three-dimensional brain circuit while not losing sight of the connections that transfer information. One of our favorite species to work with are flies. They have about 100,000 neurons. And one of the hopes that we can achieve in the coming years is that we can make complete maps of some small brains, like flies and fish, small enough that we can know when we're done. Can we actually simulate them in a computer and see them navigate, fly around, evade predators, find things to eat, and so forth. So we're already finding that we can expand and image an entire fly brain. And flies, of course, can do many things that even advanced computers cannot. So I think that this will help us really design new kinds of robotics and new kinds of strategy for interfacing to the world if we understand how the brain does it so effortlessly. And with such low power, too, I should point out. The human brain only uses about 20 watts of power. Compare that to the city block size, behemoths that you hear about in the news all the time. OK, so to summarize this first part, we've discovered that you can evenly expand brains 1,000 times or more in volume. We discovered that it's even all the way down to the resolution of nearly single molecules. And so we can really, for the first time, image large-scale 3D structures, like entire small brains and parts of large brains, with the resolution needed, we think, to make computational models of how the brain works. And I should point out that this is a very simple procedure. So people all over the world are using it. It's been applied to bacteria, to viruses, to parasites. We're starting to work with foundations like Bill Gates' foundation. Because imagine that you could take a bacterium or a virus and blow it up and then take a picture on your phone. Could you make the diagnosis of disease into a zero-cost endeavor? A few pennies of baby diaper material and cameras that are already nearly free. So that's part one. Can we image? Now, as must be all too obvious, this only works on preserved specimens, which is a euphemism for dead specimens. And so we need to be able to do different things to look at the high-speed dynamics of the brain when it's alive. So part two I want to tell you about is optogenetics. This is the way we control the brain using pulses of light. Now, as I mentioned earlier, brain cells compute with electrical pulses. How do you control them? Well, what if we had a way to convert light into electricity? And then you could aim the light at the brain cells. To do that, you need to install basically solar panels in brain cells. And then you could shine light on them and turn them on or off. And the brain doesn't feel pain, so you can bring optical fibers, not unlike those that wire up the internet, directly into the brain. And you can beam light into these deep structures. The hard part, of course, is how we're going to make little tiny solar panels. It turns out that we're really lucky. The natural world solved the problem for us. So all over the tree of life, you can find these molecules from species like this green algae that convert light into electrical signals. This green algae has two flagellae, those hair-like things that stick out. And that brown spot in the middle is an eye spot, which controls the swimming. And if you zoom in, what people found about 20, 30 years ago is that there are proteins in this eye spot that convert light into electrical signals. You hit it with light, it opens up a pore, and ions, charged particles, will flow across. So it's like a little tiny, effectively, solar panel, right? We have a little device here that will convert light into an electrical signal. So if the first part of this talk was about using mechanical engineering at the nanoscale, now we're doing electrical engineering at the nanoscale. So it turns out we were very lucky. This protein was encoded by a small gene, and we could take the gene out of this algae and deliver it into the brain using a gene therapy vector, like a virus or some other method. There's lots and lots of them out there. And this is where serendipity part two struck. The brain cells, amazingly, could manufacture this protein and install them on the membrane of the brain cell. And so all over the brain cell, we have proteins that convert light into electricity. And then serendipitous part three kicked in. These molecules, it turned out, were safe, even though neurons are really fragile. And they were fast and high amplitude enough that when you shine light on them, they would be activated. And they would fire electrical pulses, not unlike electrical pulses that are happening in your brain right now. So what can we do with this? Well, we can take these molecules and put them in some brain cells and not others by using tricks from the field of gene therapy. And I don't have time to go into that for this talk, but there's all sorts of different viruses you can use, different genetic regulation strategies you can use. So for example, you can take this molecule, which you call an opsin, and you can put it just into some cells like these little star-shaped cells that are called basket cells. And then what you can do, my collaborator here at MIT, BWSI, did just this. You can shine a laser light down an optical fiber into the brain and drive these cells. So Liwei, amongst other things, is an expert on Alzheimer's disease. And their group used our technology and found that if you drive the brain at a special magical frequency, 40 times a second, then the brain's immune system turns on, and it'll clean up in mice that are engineered to get a mouse form of Alzheimer's disease. It'll go around and clean up the debris or junk in the Alzheimer's brain. So that's pretty cool. You can control brain activity and clean up the most intractable brain disease we know about. It has a 100% failure rate for any treatment known so far. And then Emery Brown here at MIT, who knows a lot about brain rhythms, said, what if you could do it using movies? What if you could just watch a movie, no gene therapy, no optical fiber, no implants, no surgery? What if you could just see something through your eyes or hear something through your ears, and it would create the same magical frequency of 40 times a second? And the teams got together and did that too. So they took the Alzheimer's model mice, no viruses, no gene therapy, no opsins, no algae, nothing. Just had them watch a movie. And sure enough, if you had it at the magical frequency of 40 hertz, the brain's immune system could turn on, and these animals got better. So yeah, Liwei and I co-founded a company called Nito Therapeutics, where basically we're trying to build movies to treat Alzheimer's. So if we can do that, that would be a pretty major contribution. But of course, many things fail to go from mice to humans, so we'll see how it goes. What about other things? Psychiatry, addiction, learning. I'll give you just one example, because we've given these molecules to thousands and thousands of groups. I think many of us have heard about dopamine neurons. The popular press often calls them the pleasure center of the brain. But they're a lot more than that. They're involved with addiction, they're involved with learning, they're involved with Parkinson's disease. Anyway, Chris Fiorilla's group wanted to figure out if you activate these brain cells, what would happen? And so they took our molecule and put them into the dopaminergic neurons of mice, implanted an optical fiber, connected to a blue laser, and they devised a very simple behavioral task. If mice go to one point in this little box and poke their nose into a little sensor, they earn a pulse of blue light. If they go to the other part of the box, they get nothing. And so here's what they found. Here's the mouse, pokes its nose, gets a pulse of blue light, does it again, gets another pulse of blue light. I think you're seeing the pattern here. The mouse is basically working for light. So this allowed Chris to prove that activating this set of cells was enough to make the brain do more of what it was just doing, and to do it in a very clean, unambiguous way. We found lots of other molecules. We found molecules that respond to different colors of light. We found molecules that do the opposite of turning neurons on. We found neurons that will pump ions the other way, so we can turn neurons off. Imagine if we take this kind of molecule and put it in, and then you could shine light on a piece of the brain and turn off the tremors of Parkinson's disease, or turn off an epileptic seizure. And so lots of people are now using neural silencers that we've invented, as well as the neural activators. So to summarize the second part of the talk, the natural world we found gave us these molecules that just out of the box, ready to go, basically no modification needed, allow us to turn neurons off, like the two columns on the left here, or to turn neurons on, like the column on the right. And so if you activate brain cells, you can figure out, well, how do you induce patterns that can make the brain better, to heal the brain? Or can you turn off the brain and cancel out a pathological pattern of activity? So that's part two. We can control the brain with light. The last thing I want to tell you about is, can we watch the brain in action? So this is hard, because the natural world didn't make these for us. So we thought, if nature didn't evolve these for us, can we evolve them in the laboratory? And so we built robots that can do evolution in the lab. Here's how it works. Suppose we have a gene, and the way evolution works in the wild is you get lots of mutations of that gene, and some of them get passed on to descendants, and others disappear if animals die out. And over many cycles of reproduction and selection, you get genes that have these functions that help with survival. So we thought, what if we built a robot that would evolve a molecule in the lab? Take a gene, make many, many, many mutants of it, put the mutants into cells, and that's what you see in this cartoon. In the upper left, we're putting mammalian cells, actually human cells, in a dish, and each cell gets one mutant. And then what you can see in the lower left here is we take pictures of those cells with a microscope. And some cells will have better genes, some cells will have worse genes. And if we do this many, many cycles, we can evolve the molecules in whatever direction we want. We can make a bright, fast, glowing indicator of when a brain cell is active, for example. So then we come in with a robotic arm, and we suck out the cells, and therefore the mutants in them that we like. And we can harvest them and put them into a sequencer of genes. And so indeed, we can evolve large numbers of molecules. And we can make a glowing indicator of when neurons are active. So here's some data collected by Shuo Han's group at Boston University. And it shows you what our molecule does. We took a molecule that we thought could be evolved as such. We made millions of mutants, put them into cells. Some glowed, some didn't. And we picked out with the robotic arm the ones that glowed the best. And so what you can see here from the mouse brain are images that look a lot like electrical pulses that you would see with an electrode, a wire. Except these are being now taken with a camera. So we can see large numbers of brain cells being active, despite the fact that we're not putting anything surgically into the brain here. So to summarize this third part, we really would like to be able to watch the activity of the brain optically, so we don't have to touch it, and we can see large volumes with a camera. And it seems that we have that ability now, and we're continuing to make more and more molecules with our robot. And so my hope is the following. We can watch the brain in action while it's computing, during a decision, a sensation, a behavior. That's the bottom part. We're going to run this slide in reverse. Then in the middle, we can perturb using our opsins, or optogenetic tools, as we call them, opto for light and genetic because it's a gene, and perturb the brain cell's activity in order to see what those cells really do. And then finally, at the end of the day, we can expand the brain and figure out how the wires are connected to make a map of the brain. Our hope then is we can use the principles of computer science to stitch these data sets together to make a real dynamical systems model of the brain. Can we actually predict what the brain would do next? And we're starting with small brains, like worms and fish and flies. This is some data we're collecting right now here at MIT on worms that have only 302 neurons. So if we can map that, maybe that paves the way for the next step. We're also collecting data from fish. These are all just voltage imaging data. Fish have about 100,000 neurons, so maybe 3 orders of magnitude bigger than a worm. And then mice are 3 orders of magnitude bigger than a fish. And a human is 3 orders of magnitude more brain cells than a mouse. So at the end of the day, could we actually start to create models of the human mind, where you could actually simulate the processes that lead to thinking and feeling? And also, not incidentally, to try to tackle these diseases that affect so many people. So I think it might be fun to end there and take questions. I've heard lots and lots of stuff, so I literally have hundreds of slides. But it's more fun to know what you're all interested in, because this, we cover everything from robotics to evolution to optics and everything in between. But I think I might end on the most important slide first, which is this is all a very omnidisciplinary endeavor. We're bringing together physicists and chemists and electrical engineers and computer scientists and roboticists and all sorts of people to this grand quest of solving the brain. And so on this slide at the top, we have people in our group and our alumni who generated the stuff that I talked about. And then in the middle is an even larger set of people, which are the different leaders of research groups all over the world that we work with. It's really fun because we're applying every part of science and engineering to this problem. So let's end there and take questions. And then if there are resonant themes, I have lots of other things that we could talk about. Thank you very much. Yes, good.
Have they begun clinical trials to test optogenetics or not?
Yeah, so the question was, have there been clinical trials to test optogenetics? There actually have begun clinical trials. So there are a couple trials for blindness, where people lost the photoreceptors in their eyes. And by putting the optogenetic molecules into the spared cells of the eye, people, it's hoped, will be able to see again. But honestly, being in gene therapy, it's an expensive and slow and difficult process. And so I'm a big fan of this methodology that we explored here for Alzheimer's, where we develop a precision technology like optogenetics to study the brain. We then make a discovery. In this case, Leeway's group discovered this pattern of activity that could heal, at least in mice, an Alzheimer's-like state. And then the third step is the design stage. Let's build a non-invasive, dirt-cheap, practical way of getting it into the world. And that's where Emory came in, and the team then designed these movies that flicker at 40 times a second. And those we've begun human trials already. We hope to enroll several hundred people by the end of this year. And who knows, we'll hopefully unblind and know how it goes sometime next year or something. Yeah.
So you mentioned using this technology to simulate human brains, right? So my question is, what do you think would be the implications on society once we do actually have this technology functioning on humans, and we could create humans that live entirely in virtual space that could function on on much faster timescales and potentially be immortal or susceptible to cyber attacks?
Yeah, so it's a great question of whether, it's pretty clear, I think, from the kinds of data we're collecting from small brains, that maybe it'll be feasible, I don't know, maybe in 5 or 10 years, perhaps. It's always dangerous to predict, but let's throw it out there, huh? Maybe in 5 or 10 years we could simulate a small brain. And then I think it's very easy to argue it's just a matter of scale to go to larger brains. I think the big question, the elephant in the room, is would it feel like you if we were to simulate a circuit? We don't yet have any way to measure consciousness. Does somebody have feelings? Does somebody have thoughts? I mean, you don't know for sure if I'm actually having feelings while I say these things, right? I could be a very accurate simulation of me, and my real body's somewhere in prison in the mountain range in the middle of nowhere. I hope not. If it were, I would be programmed not to say that. But yeah, I think that's one of the biggest questions is, can we measure consciousness? Can we measure when somebody's aware or alert? And that's something we're actually starting to try to figure out how to do. So I didn't put into the deck of slides that I showed so far, but we have been trying to work on ways to stimulate the human brain non-invasively. So could you drive a feeling or a sensation or a smell or a memory, and what does it feel like? So a year ago, we published a paper where we discovered the brain kind of acts like an old-fashioned AM radio. The brain is known as to be a so-called nonlinear low-pass system. And that's exactly what an AM radio is. So the way an AM radio works is you beam the radio waves across the city, and then the radio receiver will have this nonlinear low-pass property, and that's how you extract the voices or the music or whatever, right? And so we've known for 100 years that brain cells are like that as well. And so if you put one high frequency over here and a second high frequency over here, the brain just ignores them, the same way you can't hear radio waves going across the city. But where they collide, the nonlinear low-pass effect will actually extract the difference between them. So if the difference is something small, then you get something that the brain could follow. And that's exactly how a radio extracts your voice, or the voice of the broadcaster. So I would like to start working on this. It's early days. We're just now starting to get data from human subjects. And it's just a couple people. But if you could stimulate the brain in a 3-dimensional pattern, could you play back activity of the brain? And then we could ask the people, well, what did that feel like? So that's one answer to your question. The other answer to your question is we should get people talking about it sooner than later. So one thing I was talking with some friends a couple months back, that there is no global neuroethics conference that brings together people from different cultures, people from different spiritual beliefs, different scientists, lawyers, doctors, engineers, entrepreneurs, and so forth. And now that companies like Facebook are getting into brain scanning, they have 80 people building brain scanners now, right? I think there's some urgency, right? Questions of privacy, questions of identity are on everybody's mind. And of course, if you're scanning somebody's brain, that certainly is as close to home as it gets, at least to our current knowledge of science. So we should talk about it. So we just announced, actually, although it's not a very public announcement, we're still working on making a more public announcement, that in 2020, I think we're going to try to have a global neuroethics conference and bring together especially industry and academia and governments and others. Because right now it's been kind of more of a balkanized conversation. And in other words, can we talk about it? And that's the first step, because if we can't talk about it, then we're really in trouble. So yeah, great questions. Other questions? Yeah?
So there's expansion microscopy in single cell arrays. Do you do location-specific transcriptome stuff? Is there a strategy for that?
Absolutely. Yes. So Let's see here. Okay, here's the 425 slide version of the deck, where I have lots of backup slides for hopefully any question you can come up with, maybe not. Okay. Yeah, so the transcriptome is the expression of the genome. Most of our cells, basically the same genome, more or less, but different parts of the genome are on or off, and that makes cells different. So a couple years ago, we actually did show that we could expand a piece of brain tissue and then label the expressed genes through a process called in situ hybridization. Basically, we expand the thing and then bring in glowing tags that will bind to those and allow us to visualize their identity and location. And recently, a group at Harvard has shown that you could expand and then label these expressed genes with sort of cleverly coded probes that you can apply one after the other. So imagine each probe round, you can apply something, and there's a 0 or 1. And you apply it again, you get another 0 or 1. With 10 rounds, you have 2 to the 10th power, or 1,000 different possible tags. That's almost a pretty good chunk of the genome, right? It's 15 rounds, you get the entire genome. Anyway, she calls this MERFish. This is from Xiaowei Zhuang's group at Harvard. And she showed that if you expand tissues first, you decrowd the molecules from each other, it becomes easier to follow them over these many cycles of hybridization, and you can get order of magnitude improvements in the yields. So yes, that was the first description, I think, of how expansion can really help with whole or nearly whole or partial transcriptome analysis. Great question. Well, so they're expanding, and then they're doing hybridization right in the cell. So you can do all your assays right there in the expanded cell. So it is effectively single cell, or even better, because you can see exactly where the transcript was in the cell. Is it in the nucleus? Is it outside the nucleus? Brain cells have one nucleus, but 1,000 connections. You might want to regulate genes in a spatial fashion that's very, very nuanced. And in fact, we're starting to get some data that suggests that is the case. So, yeah, great question. Other questions? Yes?
Is there any coding in your job?
Any what?
Is there any coding in your job? Because you mentioned that there were a ton of people.
Any coding in my job?
Yeah, you mentioned a ton of specialties in your field. So how much do you guys get in that?
Yeah, so we routinely do coding just to analyze all the data we're getting. I mean, these data sets are enormous, and we have to analyze them in many ways. Are we ready to build simulations of the brain? Not yet. I started out as an electrical engineer, computer science type person, and chemist, and did some physics as well. I ended up kind of focusing on the chemistry side for the last 10, 15 years, in part because we didn't have data that we wanted. Without the data, how do we actually simulate what we want? But in the coming 5 or 10 years, my hope is we're going to converge back into the computer science side as well. So actually last fall, we published our first complete computer science paper, where we took, we simulated expanded brains and then worked out machine learning algorithms that would let us trace the neural circuits with great accuracy. So we're getting into that now. Yeah. I'll just go through the room, I guess, instead of kind of being ad hoc. Yes?
In the next 10 years or 15 years or so, what do you see as the big breakthroughs that you might see?
Yeah. So I'll make 2 predictions for the next 10 years. One basic science and one clinical. I think the clinical one is kind of I already alluded to earlier. These intractable diseases like Alzheimer's that have a 0% treatment rate, basically. I think through studying the brain, we're going to understand how to tackle them. So this whole invent a basic science technology, make a discovery about the nature of a disease, then design a practical treatment, like we did here in this project that Li Wei's group led. I think that's going to become more and more common. So we're already talking with people how to apply this kind of invent, discover, design strategy to brain cancers, to sleep disorders. How do we help people sleep better? To depression and anxiety disorders, and to Parkinson's and epilepsy and beyond. My second prediction is that if we can simulate an entire small brain, that would vastly help us in understanding how to build better computer algorithms, and that will revolutionize AI and robotics. Even solving a small brain of a worm or a fly. I mean, again, these are critters that on an almost 0 energy budget can do amazing things. And the versatility and their ability to learn quickly, for example. That's one of the things that machine learning struggles with, is how do you learn very, very rapidly? Well, it's clear that brain circuits in their wiring and in their rules have some information that we haven't quite figured out yet that allows them to learn rapidly and to do things that are difficult to do, like generalize from one problem to another, but which brains can often do quite easily. Yes?
did put the glowing, I guess. So how do you, can you explain the glowing thing, like with the glowing, like or whatever cell?
The last part in the glowing voltage?
How do you with like, I don't know, like where you manufacture that?
Okay. Yeah, let's go through that with more detail then. I never know what people are interested in, so I was trying to give a high-level overview of things. Okay, so we wanted to make a fluorescent voltage indicator. What that means is we shined orange light or red light, and they would glow with a farther red-shifted color of light. So maybe we shine on 630 nanometer red light, and it would emit 660 nanometer red light. So that's our goal. Why red light? It's because red light goes deep in the body, right? It doesn't get absorbed by blood. That's why blood is red. Whereas if you use blue light or green light, like I don't know what the color this laser pointer is, it's a very bright green laser pointer, and you can't even see it through my hand, right? But if this was a much weaker red laser pointer, I don't know if we have a demo here, but you could see it through my hand easily, right? I think we've all done things like that when we were little kids. Okay, so what we do is we take a candidate gene, which we suspected could be mutated into a glowing voltage indicator. And that was for two reasons. One is that a group at Harvard had discovered this basic function many years back and said, look, it's a weakly glowing fluorescent voltage indicator. Too bad it's weakly glowing. And then we thought, let's mutate it and screen for ones that are brighter and that they localize well to the membranes of the cells and so forth. So we make a lot of mutants and put them into the cells using a process called transfection. Every mammalian cell gets one mutant. We let them express the gene for a little while. That's what these little glowing things are supposed to be. And then we scan a plate of these cells with a microscope. So we're going to shine red light and then image with red, and then look at the redder light that comes back. That's fluorescence. And then if a cell is really bright, we'll grab it with this little suction robotic arm. Basically applies a vacuum and sucks up the cell. And then we sequence the genes that were in the cell, and that tells us what the mutant was. And then we can do this many, many rounds. Did that help? What do we do to get the experiment to work at the end? Okay, so at the end of the day, we have a gene that if you put into a cell, the cell will manufacture this glowing protein. So we take that gene, put it into a gene therapy vector, just like we did for the optogenetic tools I mentioned earlier, inject the gene therapy vector or virus into the brain, and then the brain cells manufacture the glowing protein.
So can you like stick it in a sperm cell and make like a glowing mouse?
You can do all sorts of stuff. Yeah. People are doing that very kind of thing. So actually, for these fish that I mentioned, or for these worms, what they did was to inject these into the eggs. I've never done this myself. They're done by our collaborators. And then the offspring grew up and turned into these glowing, in this case, worm. Yeah. And there's actually a person here who's built a robot that does that automatically. So all the worms are lined up and just inject them, pip, 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 and then they give birth, and you have a worm that glows. So you can make these in a very high throughput fashion. Yeah. Other questions? Yep.
What mechanism exactly makes like the light wavelength increase? Like you said,
Say it again.
you get like redder light, you shine red light on that gene, or some, on those cells with that gene. What makes like the light wavelength increase?
Yeah. Well, there's a process called fluorescence. So you have an electron, and it's at a certain energy level, and if you shine light onto it, it gets excited. And then there's some loss of energy from vibration and just being in the world and interacting with the thermal bath of our universe. And so it relaxes a little bit, and then it falls back down and emits a redder wavelength. So the classical protein, green fluorescent protein, you shine blue light on it, excite the electron, it relaxes a little bit due to the interactions with the environment, and then emits green light. But as I mentioned earlier, we don't want to use blue and green because those get really absorbed by the blood. So instead we use one color of red, and then it vibrates a little bit, and then emits a longer wavelength of red. And so this is, like glow-in-the-dark stickers have like a really, really, really long version of this that involves some other states that delay it out. So it's like a really fast glow-in-the-dark sticker. Great questions. In the back.
I was at a conference and learned about this technology to help diagnose people in like a vegetative state with the brain-computer interface. That technology relates like the AM radio nature of the brain and like read how they're.
So they're monitoring the brain for that?
Yeah, like determine.
Sure. Well, suppose you can monitor the brain and see that a certain part of the brain is underactive. And suppose we then go to that very same patient and try to jazz up that area and increase the activity. Maybe we could try to reverse the condition. And in fact, there's a person named Nicholas Schiff who tried that with implants. He took patients who were in a persistent vegetative state or a coma, and he implanted electrodes into the thalamus in the middle of the brain. And he showed that he could partially reboot the brain. These patients would respond to noises, they could eat. Before they couldn't even move or respond to anything. Now imagine we could do that without surgery. So that's one thing that actually with Emery Brown, the very same person who we collaborated with on the Alzheimer's study, he's an expert in anesthesia, and he's shown in the past that if you activate certain parts of the brain, you can actually wake up a rat anyway, even though it's anesthetized, which is kind of weird, right? Imagine something that's fully out and you can wake them up. So maybe there are some circuits in the brain that we can send our energy into and reactivate them without any kind of implant. Yep.
Technology to cure brain disorders and diseases. But do you think there's a possibility anyone will use this type of technology to look at improving the brain, brain as a whole, is it functionality-wise?
Yeah, augmentation. Yeah, so I think there's sort of a natural trajectory of human augmentation, where technologies start often in medicine, because that's where the need to risk ratio is the highest, right? But what happens over time is we see that technologies, as they're proven to be safe and effective, they do broaden in usage. And technologies that are not safe and effective, they shrink in usage. So we've seen this already in brain technology over the years. Antidepressants, attention deficit disorder drugs started out for very narrow populations of use, and then over the years, the usage has spread. And so I think it's sort of a natural time constant of maybe a couple decades where you see, society changing its attitudes towards something. And so this is not a judgment that it's right, not a judgment that it's wrong. It's just an observation of what we see in the world. So it goes back to an earlier question about, what do we want? Do we want to augment our brains? In what ways? And that's, again, where I think we need to launch a global conversation about what do we want to do, what do we not want to do. And something like that happened for genetic editing, right? 1975, in Asilomar, California, Paul Berg, who won the Nobel Prize for his work on restriction enzymes, called a conference and invited the press and scientists and doctors and everybody into this little campground in Monterey Bay in California. And for days, they talked about what do they want to do, what do they not want to do. And in the half century since, we've had all sorts of insulin and growth factors and vaccines and antibiotics and all sorts of stuff that emerged from this, and very few negatives, because the group decided to self-govern and to put out rules about what they wanted to do and how they would decide, right? So at MIT, if you want to do anything with DNA, you have to go through a committee. You say, here's what we want to do, here's what the benefits are, and here's what the risks are. And then a committee of people, including students and professors and doctors and lawyers and other people, will get together and talk about it. And I think we need to do something like that for the brain.
For example, like CRISPR, what do you do with people who decide not to ignore those rules?
That's something I'm worried about. I mean, there already are people who are doing DIY brain stimulation, where they're building electrical circuits and trying to stimulate their brains. I think this is not a good idea, because we don't know the long-term effects of doing that. What if somebody does that and then 6 months later, or a year later, most of the studies on brain stimulation so far have been short-term, right? With volunteers who are stimulated for half an hour or something. I think that is a problem, and that's another reason why I want people to talk about it, because right now it's almost a taboo topic. I was on a panel once where the chair of the panel asked the audience how many people there had tried some kind of brain stimulant or something like that, and nobody raised their hand. And then he announced that he had done an anonymous poll, and something like 10% or some non-zero number of the people should have raised their hand. And so I think that's one reason why I want to help start a global discussion about this. If people can't talk about it, then we're really doomed. Okay. Yep.
What are some of the inhibitors to generating computational models from data you get from extension microscopy? And is there any notion of how sensitive those models would be to inaccuracies in those data or models?
Yeah. Okay, how do we make the models? Well, it's early days, but here's an idea. Suppose that we did the voltage imaging and then we did the optogenetic control, and then we did the expansion. So at the end of the day, we have a network that we see with the expansion. I don't know, let's make this a sensory neuron. Let's make this a motor neuron that sends projection to the muscles, right? So these are the cells, and those are the connections. Well, obviously there's going to be a lot of unknowns in the network, right? We won't know how strong or weak a connection is. We won't know whether a connection has a plus sign or a minus sign added. But we can assign variables to these. So maybe we have a strength, SI, and a speed, kinetics, KI. And the strength, of course, has a sign associated with it as well. And then maybe we have some internal variables, VI, inside the cell that tell whether the cell responds strongly or weakly to an input. Now, we don't know what those are. But can we use inference, statistical inference methods like machine learning strategies to infer what those are based upon these two data sets, the voltage that we watched and the optogenetic control that we perturbed? So suppose that during the course of the experiment, I delivered light to stimulate this cell, and I saw this cell had a response of 3 units. I'm just making this up. And then we stimulated this cell, and we saw that this cell had a response of 2 units. We could maybe start to infer the strength of these connections. So that's how I'm thinking about it right now. Get the map in the third part of the experiment. We have the dynamics from the first 2 parts of the experiment. Let's infer the hidden variables from the first 2 parts. And some, using simulated data and some back-of-the-envelope stuff that we're doing, it seems like it's not totally implausible. It seems like if the dimensionality of the data can confront the dimensionality of the space that you want to fit, of course, modulo the noise that you have in the system, then maybe there's a shot on goal. So that's what we'd like to do in the future. Other questions? All right, I think I covered the whole room. So anybody who raised their hand out of sync? Yes?
What was the name of the organism? You used the one that you got into the ion beam?
The organism that we are studying?
Yeah, the one you used for optogen.
So the algae that we got the activator from is called Chlamydomonas. And then the silencers come from a area of biology called archaea, which are kind of like bacteria, but also different. The one that we use a lot is from the genus Halorubrum, for example. So these come from different parts of the Earth, different algae, different bacteria, different archaea, even funguses from all over the world. And then we analyze the genomes and then put the genes into cells, not unlike what we did with the robot, but we're doing it by hand, and then look at what the cells make. Yeah, so it's a lot of fun because you discover all sorts of weird stuff out there in the wild. Other questions? Yeah.
Thank you so much. That was a very interesting talk. I was wondering if do you have any resources you could recommend, like papers and books, to learn more about neurotechnology in general?
Neurotechnology in general. Wow, that's a good question.
Or just more about your work.
Okay. Yeah, neurotechnology in general. I keep getting asked to write a book, and I have turned it down so far because by the time the book was done, the whole field would be different. So what I've been trying to do, because it's such a fast-moving field, it's one of the fast-moving fields in all of science and engineering, for better or worse. Mostly better, because it's a lot of fun as a result. But so my group's website, synthneuro.org, has a bunch of video tutorials and explanatory web pages and so forth. We've been using the internet to update in real time rather than try to write books that will sit there obsolete with outdated information gathering dust. And then recently I've started using Twitter as a way of broadcasting information. And there's a lot of neurotechnology is so fast that Twitter is a great way to find what some crazy new tool is from the middle of nowhere. So that's become really, really valuable as well. So I'm eBoyden3 on Twitter, and that's a great way to find neurotech information in real time. I've discovered many interesting things on Twitter long before, sometimes years before they come out in publications. It's a very active community in the neurotech space. Other questions? How are we doing on time? I do have 400 more slides I could show, but it looks like we're out of time, so we'll end there.